Howdy, our names are Cassandra Fernandez and Alondra Curiel, and today we're gonna present our microscope and staining lab.
In this lab experiment, we use smear preparation and simple staining to observe bacteria under the microscope. Simple staining with methylene blue increased contrast, making it easier to see the size, shape, and arrangement of cells. This allows us to study cultures of Staphylococcus aureus, Bacillus cereus, and Salmonella typhimurium more clearly.
During this lab, we used methylene blue, a, st- a sterile inoculating loop, a microscope, distilled water, bacteria, and the hot plate.
In the procedure, we got all our items that we needed. We first had to clean the our glass slides with a specific paper, a cleaning paper that we were given in the lab. And then we used a red pencil or color to make a circle in the back, write our group names, etc. And we then had to uh put a small drop of DI water and mix in our bacteria that we got from the sterilized loop. And this procedure we just learned how to properly um collect bacteria from our media and transferring it onto a slide and making sure it was spread around a size of a nickel, and it was placed to dry on the hot plate. And once they were dry, we were able to heat fix them by using the Bunsen burner.
For the simple staining process, once the slides have dried, we put the slide to stain on the staining tray after they had been heat fixed. Then we place the slide on the staining tray. Then we flooded the slide with methylene blue stain. After we stained it with methylene blue, we let it sit for 1 minute before we stained it with DI water. After it was rinsed with DA water, we dried it effectively and then we placed it under the microscope. However, we did not use the 100x during this lab, so we did not need the oil immersed on the slide.
For our results in these series, we mostly saw rod shaped, uh, or also known as bacilli. We saw a lot of rods scattered singly. Some rods were in short chains, and there was noticeable dense clumps of cells in a couple spots on the side. That's likely where the smear dried thicker, or like we just put a little bit too much. And it shows the rods with some clusterings and short chains. For the SRs, the prep or the kind of the slide prep, it looks very thick and uneven with a lot of background staining and large dark aggregates. This just could be because we added way too much bacteria. Because the sample is so dense, it's difficult to see the individual shapes, but there are hints of small cocci and rods mixed within the background debris. So it is too dense to confidently interpret it due to the amount of bacteria that's on it. For S. Timbimurium, we could see the cells are round or clock-eye shaped, and you can see several long chains and some small clusters and pairs like scattered around. And these were all blue. They didn't have a specific gram negative or positive distinction, aka purple or pink, because it didn't have any of the other thing. It was only with methylene blue, so we could not tell if they were gram negative or positive.
The staining and microscopic test showed three different outcomes: bacilli scattered singly and in short chains with some clumping, an overly thick smear that obscured morphology, and cocci in chains consistent with streptococcus-like patterns. Slides were heat fixed, stained with methylene blue for 1 minute, rinsed, dried, and viewed under the importance of proper smear preparation and staining to clearly observe bacterial shape and arrangement.
Thank you for watching. I hope you enjoyed our lab report video.